and this paper fish, Medaka fish, are able to recognize each other based on things such as visual cues, odors, and other chemical cues. So it has been assumed that familiarization can affect mating preference, but just the neurobiology and the molecular biology behind the pathways to create this preference has been largely unknown. So Medaka mating. Medaka are this very small little fish that is used a lot of times in molecular biology experiments. So there are several steps for Medaka courtship. The male will approach the female, and if she responds to his behavior and is being receptive, they mate. However, if she rejects the courtship behaviors, he moves on and displays his courtship again to another female. And this is measured by latency, and that is the time between the first male demonstrating his behavioral courtship displays and the time it takes for them to mate. So they did many different hypotheses. So I'm just going to kind of lead you through all of them. I generalized a lot of the experiments since many of them were testing for the same thing. So females will have shorter latency periods with familiar males. The shorter the latency period, the higher the female's receptivity to the male. And then males will not demonstrate the same preference for visual familiarization as strongly as females. So in other species, females are the ones that are really kind of picking the mates for them, and males are just kind of showing off for whoever they can, and if they can mate, great. If not, they're going to move on to the next female. To test kind of how the Medaka fish use familiarization, they performed an experiment to remove eyes to test if visually, that is the most important way for them to kind of recognize each other. And then neural and molecular factors will affect female mating preference by either suppressing or encouraging this receptivity. So different methods that was used. They bred Medaka fish in plastic aquariums, and they bred a lot. They bred hundreds of Medaka fish for many of these experiments. They determined the phenotypic sex from the secondary sex characteristics such as the dorsal and anal fin shape and the length. They did many behavioral assays to determine visual familiarization versus none. If a female is visually familiarized with one male and it mates with another, how does that affect latency, and vice versa with males. They removed female eyes by cutting off nerves. They did cell transplantation. They performed a luciferase assay, laser ablation, and then finally a paternity assay. So this first image, group 1 and group 2. In group 1, the females and males were in the same tank, and they were separated by a glass wall. So the female and the male could very clearly see each other, and this is so they got visually familiarized. Group 2, same thing like Medaka fish, but they weren't getting visually familiarized. There was a barricade, like a black wall in between where the females were kept and where the males were kept. Group 3, a female was familiarized with a male, but then was mated with an unfamiliar male. Group 4, a male and a female in pairs were visually familiarized, and then to measure latency, they actually the experimenters switched the partners of the 2 and 3 were very similar, so that kind of led to the conclusion that female preference was important, and especially visually, and the fact that they were being mated with an unfamiliar male really increased the latency because the male had to perform more courtship behaviors. Group 5, this and a couple other experiments, the males really didn't have any changes in receptivity, so it's really only the females that are displaying differing levels. This figure, figure C, is the experiment where they tested whether it is visual familiarization for sure. So the sham group is, they damaged the eyes, just above the eyes, the nerves above the eyes, so they couldn't see that well, but they could still see, they could still recognize their partner. And group 1, which is still the visual familiarization, was still significantly lower than group 2. The single, only one eye was removed, so they could still see their partner, and still group 1 had shortened latency periods in group 2. However, when both of the eyes were removed, the latency periods between groups 1 and 2, for the most part, are approximately equal. So when the female cannot see the male that she was familiarized with, the eyes were very important in determining that visual familiarization was the big key factor. They also determined that a couple receptors in the cell membrane were kind of playing a role in how the fish responded. So in this, they noticed that the CXCR7 receptor was seemed to be correlating with a lower latency period. So the CXCR7 and CXCR4, they're chemokine receptors. They're in the cell membrane. They respond to specific signals, and through these experiments, it was determined that they actually enhance female receptivity. So they make females more responsive to the male courtship. To test kind of where these are, the CXCR7 and CXCR4 play a role in the GnRH3 neurons, and to kind of figure out how that plays a role, they tested the 3 different regions these GnRH3 neurons are located in the brain. So the POA is the preoptic area. This is the terminal nerve area and the midbrain region. And they determined that only the terminal nerve area was involved in behavior that is related to mating. So the terminal nerve GnRH neuron. In order to learn more about it, they performed laser ablation, so they cut off the receptivity of these neurons and performed an experiment to where, the group 1 and 2 in the sham still were able to use these neurons, but these had been removed, so they could not, the receptors could not respond to any signals, and they noticed that the latency period was significantly decreased in this. So they performed another test, and they created a mutant of specifically a GnRH3 mutant. So this showed that there is another reduced activity in the cell membrane, and this mutant was the GnRH3 S27P/S27P. So the females with this mutant showed that instead of having enhanced receptivity, it was actually suppressed. This didn't affect males at all, it was just females. So the CXCR made the females way more receptive, way more likely to mate. This mutant actually decreased the likelihood that they would mate. And it was noticed that the GnRH3 peptides, so that neuron is required for this mutant to work, and is required for the neurons in the terminal nerve to function. Finally, they did a paternity test assay. So basically, there was one female and then they had two males. One male was a wild-type male, and the other was with a luciferase assay, had a green protein, fluorescent protein. So this was done to make it easier to test who the offspring belonged to. So in step 1, the female was visually familiarized with both males, and as she got pregnant and they were able to test the offspring, they noticed that the TG, the one with the green protein, had a higher likelihood of mating with the female. But then in set 2 and 3, where only one of the males was mated with the female and had visual familiarization, each of them had the same likelihood of mating with her had they been visually familiarized just then. So she was more likely to respond to the familiar male than the unfamiliar male, whether or not it was the wild type or the mutant. So once again, originally the male who was expressing the green fluorescent protein had a higher chance of winning. But as more tests were done and they were able to test visual familiarization, it kind of evened out. So visual familiarization ended up being very important. However, if a wild type female was visually familiarized with one male, but then was in with an unfamiliar male, and this unfamiliar male was of a larger body size than the familiar one, there was a higher likelihood that she was going to mate with the larger unfamiliar male. So that kind of shows that even though visual familiarization is very important, receptivity also plays a key role in determining body size, and she's going to be more receptive if the male is kind of stronger and bigger as compared to the male that she is able to recognize. The bottom two figures are just showing once again the two different mutants that were used. So the CXCR7 mutant, the familiar male and the unfamiliar male each had a very similar chance of mating. So the female, the receptivity increased, so the female is more likely to mate with either one, no matter if she was visually familiarized before the mating occurred or not. However, the opposite occurs in the GRH3 mutant. It actually suppressed the receptivity, and these females ended up being almost a lot less likely to mate at all. So, conclusions. The CXCR7 and CXCR4 mutations enhance female receptivity, and on the flip side, the GnRH suppressed it. And then the terminal nerve, GnRH3 neurons suppressed it, specifically the terminal nerves. The GnRH3 peptides are necessary for the terminal nerve neurons to be functional. So in order to have any effect on receptivity, the GnRH3 peptides have to be able to be received by these terminal nerve neurons. And then it is very possible that the visual familiarization itself triggers the release of the GnRH3 peptides. So just having the visual familiarization is able to enhance the receptivity. And then, to acknowledge Okuyama et al. For his article in Science